Hi everyone, my name is Giulia, and today I will present one of my latest work called Cell Gossip. We know cell communication is very important, in particular for multicellular systems. So we know that cells are not isolated entities, but they are organized in tissue, and so they communicate constantly with each other through the intercellular signaling, so signaling that happens between cells that are mediated mainly by the ligand-receptor interaction. Once the cell receives this external signal, it converts it into an internal signal through a process that is called intracellular signaling transduction mechanism. This involves the activation of signaling pathway, so a cascade of biochemical reaction that affect the cell physiological functions. So, for example, activating transcription factor that in turn regulate the expression of target genes. So we have information that travel from the receptor to transcription factor and then to the DNA with the gene expression. But this process is highly cell-specific. This means that different cells respond in different way based on its identity, role, and function within the tissue. So as we know, single-cell transcriptomics data enable us to study cell-cell communication and intracellular signaling because it provides the gene expression, so somehow the proxy of gene activity, as single-cell resolution, so capturing the cellular heterogeneity. We know that comes with different limitations, like the inherently sparse noise of the data due to the high dropout rates, and also it is very high dimensional. And this can obscure true signaling, especially in the context of intracellular signaling. Different methods have been developed for study cell-cell communication. Among the few that perform also this intracellular signaling modeling, we have that they lack in different areas. So, for example, some of them just ignore what happens between the receptor and transcription factor. Others ignore the gene expression of key molecules that mediate this intracellular transduction. And most of them perform the analysis at cell type level. These fail to capture the heterogeneity in the intracellular and transcription response. In the context instead of the sparsity and noise of the data, different denoising approach have been developed, like MAGIC, which allows to impute the smoothing the gene expression using the cell-cell network, but these ignore biological priors that we have on the interaction between genes. An exception is SCNet, which is one of the latest tools developed that perform this imputation using both cell-to-cell and gene-to-gene network, but it does not consider the influence of the external signal, so the ligand-receptor interaction. That is one that starts the flow information. So today we'll present Cell Gossip, which is designed to reconstruct this intracellular signal transduction mechanism as single-cell resolution, integrated two source information, so the single-cell transcriptomics data that provide a noisy but an informative proxy of the gene activity in each single cell, and prior information that we have on the cellular mechanism, so this intracellular signal transduction, that capture the flow information from the receptor to transcription factor to target genes. So the key core of Cell Gossip is a personalized network propagation algorithm, seeding the signal at the ligand-receptor pair, and this is done independently for each single cell, and then also perform a cell type specific analysis to identify key transcription factor and signaling pathway. But going in details, Cell Gossip model the signaling propagation as a discrete time Markov chain averaging system, so it's an iterative process, where in this case we have that D represent our directed gene-gene graph, representing the signaling pathway. So it controls how the information spread through the network, simulating like process similarly around the walk. Then we have S that represent the intensity of the signal in each node, so gene, in each cell. And these come from the gene expression. Then we have Z, that is the personalization term, that allows to seed the signal to specific input node that in our case are the receptor. So this is computed using the ligand receptor inference tool. So there is many of them that have been developed that do the cell-cell communication. And finally, we have the damping factor, which balance between the signal propagation and through the network and the restart to these input nodes. So what we obtain is this reconstructed gene activity through a pathway-aware process at single-cell resolution, and this allows to identify there are different cells that respond a different way at intracellular level. Then it performs also the analysis as cell type or cell state specific analysis to help for the subsequent analysis. For example, in this case, is integrating two complementary source information that we have on the transcription factor activity. So we have the one I call here transcription factor signaling, which is the single cell transcription factor activity reconstructed with the signaling propagation that we have seen before. So capturing what is upstream to the transcription factor. And then the TF regulon. So in this case, it's a single cell transcription factor activity, single cell resolution using the regulon-based analysis. So based on what is downstream of the transcription factor, so based on a gene expression of target genes. And of course, we can do a consensus between these two measures using, for example, Spearman correlation through the cell of a specific cell type or cell states, and this allows to identify in a robust and biological meaningful way which are the transcription factor active, because we observing both complementary source information that follow the same trend. And then also summarize information at pathway level, so summarizing the impact of this transcription factor in the signaling pathway using a weighted mean. So in this case, it's based on the consensus between the two complementary source information and weighted by the p-value coming from the correlation. Okay, so go to the validation part, which is one of the most challenging here. So here we apply cell gossip to a unique control environment where we have the transfection of the exogenous ligand receptor pair. So here we have the natural killer cells that were transfected with the CD40 ligand, the B cell that were transfected with its connector receptor, and the GVFP that we see before, transfection is used as control. So we have 4 experimental conditions. So from the fully transfected experimental condition, the control, and then the 2 intermediate where the transfection of the ligand and only of the receptor. So we start with the pathway activity analysis performed in the B cell, but across the experimental condition in this case. And among the results show that the NFK beta signaling pathway is the most highly variable pathway. And this aligns with the role of CD40 signaling in activated designaling pathway. You can see here that actually cell gossip predicts the high pathway activity for 2 experimental conditions. This is the fully transfected experimental condition, but also the receptor-only experimental condition, suggesting that maybe the expression of the receptor or only a basal level of the ligand may be sufficient to activate this intracellular signaling pathway. If we go in detail on the transcription factor associated to this signaling pathway, we have actually 2 transcription factor modules, or 2 heterodimers. This is the canonical transcription factor model. This is the non-canonical one. And here I put the TF consensus between the cell gossip reconstructed activity and the one of the regulon, so based, for example, on the coupler. And you can see that we have a high correlation with the TF regulon, in particular for the non-canonical pathway, for the 2 experimental conditions that I said before, suggesting that we are capturing the same signal, and also we are seeing that there is a different activation of the 2 transcription factor module. Then we asked if cell gossip was able to refine this cell-cell similarity between cells using the experimental condition as ground truth. In this case, we try to compare with other approaches, so using the original gene expression matrix, using the one imputed by MAGIC and SCNet, and we do the UMAC embeddings. The cells are colored by the experimental condition. And here you can see that actually cell gossip is able more to separate the experimental condition, so able to capture more this perturbation-specific response. And of course, to quantitatively measure this, we do the adjusted random index, so computing the Louvain cluster across different resolution. In this case, we can see that cell gossip outperforms the other methods. Then we wonder if cell gossip was able to capture this heterogeneity in the signaling pathway, so see if the different cells respond in different way. So we perform cluster analysis on the B cell, and it reveals 4 different subclusters of B cells. And if we look at the gene activity reconstructed in cell gossip, so map in an FK beta signaling pathway using PathView. So in this case, to help you the visualization here, the nodes are segmented into the 4 subclusters, and the map is the scaled reconstructed activity. And actually, here highlighting because it's difficult to see, but different subclusters show show a high expression of genes related to different branch of this signaling pathway. And in particular, I want to highlight also only the yellow cluster, where in this case it's actually activating 2 paths inside the NFK beta signaling pathway. And if we close look, this is actually the CD40 axis that activated 2 transcription factor module. Finally, we try to validate also the cell gossip using these recent data from Nature Cell Biology. So an ongoing validation, but here they perform this large-scale single-cell perturbation dataset across different cell lines, targeted different signaling pathway, and using the gene knockdown. But the most important thing is that they provide this mixed scale score, which is a single cell specific score that quantifies the gradient of perturbation response. So capturing both the biological variability, but also the technical variability, like the efficiency of the knockdown. So this means that a low mixed scale score, no effect on the pathway, so the pathway remains unaltered. High mixed scale score means a high efficiency of the knockdown, so the pathway should be down-regulated. So what in the paper highlighted is that as we expected, that there is an inverse relationship between the mixed scale score and the gene expression of the gene that is targeted. So we wonder also cell gossip was able to capture this trend. So I computed the Pearson correlation between the mixed scale score and the reconstructed gene activity in the genes of the targeted pathway. In this case, there is the TGF-beta pathway that is perturbed across the different cell lines. Here I'm plotting the same also for using the original gene expression. So it's the distribution of the Pearson correlation using both the original gene expression and cell gossip reconstruct activity. And you can see that in both cases we show a negative correlation. So this inverse relationship that I told before. Not only, but also cell gossip show a stronger negative correlation, because it's able to enhance and capture more this perturbation in the signaling pathway. And this is true also for other cell types, also capturing this biological variability that we have in different cell lines. So to conclude, I presented cell gossip, which is designed to reconstruct this intracellular signaling mechanism using pathway-aware personalized network propagation algorithm, seeding the signal at ligand-receptor pair. And using this approach, we are able to capture which are the intracellular signaling that is modulated by a specific ligand-receptor pair. So like in this case of the transfection CD40 axis, is also able to identify heterogeneous subcluster of cells that highlighting different activation in different parts of the same signaling pathway, outperformed other methods to identify cell-cell similarity, especially in the context, of course, of the intracellular signaling, and is able to detect this gradient or of perturbation strength using the perturb-seq dataset. So, would like to thank, of course, the author of this work, and especially James and Ivan for help in action.
Okay, going up there. We have a cluster of questions.
Some great name with the tool. I wonder whether you are considering already, or eventually you might consider, how to deal with the crosstalk of the different signaling networks, because what I understood is you're starting with the receptor and then seeing downstream to it, but maybe there is another receptor that is triggering some crosstalk and you have regions in your network with higher activity.
So in this case, actually, you can see the signal not only to a specific ligand receptor, but you can use, of course, all the set of the receptor that might be activated in the cells. And of course, this is the suggested way, because of course, what we see in the cell is not coming from one receptor. So maybe when you see the signal to some receptor, because you want to highlight that path, but actually what you see is a combination of different signaling from different receptors. So it's suggested to see the signal among all the receptors that you have in the cell.
Thanks.
Thank you very much. Very nice talk. I was wondering whether you are able to capture as well the velocity of the activation of the signaling pathways. Because here, when you look at the regulons and the activity of the pathway, you assume that everything is just up, right? But at single cell resolution, you might be also able to find basically the dynamics of the activation. Are you able to capture this type of information, or the resolution is not good enough?
No, actually, we are able to capture, and this is something that we will also highlight in the perturb-seq dataset, that maybe is also better to see this type of things. But yeah, actually, in this case, for example, we see that we are lighting these two paths, but actually that are not activated in the same way. Of course, this known canonical path is more activated, but here it's just this scale, so it's different to see. But yeah, we are capturing also this. So actually, we are somehow capturing how the signal flow inside the signaling pathway.
Just a very, very quick question, which is probably interesting lots of us. When it will be available?
Great question. Thank you. It's not available yet, but will be released when we submit the paper. So I think in a couple of months, I hope.
Okay, so I think we are done for the morning session. Thanks again, Giulia.